salient features of chromosomes. Chromosomes can be circular in shape and they can also be linear in shape. In case of prokaryotic cells, chromosomes they are circular in shape, while in case of eukaryotic cells, chromosomes are linear in shape. The number of chromosomes is specific to the species. Based on the number of chromosomes, species can also be identified. Copy number of chromosomes. Chromosomes can be haploid, diploid, or polyploid. Based on the number of chromosomes, cells they can be called as haploid, diploid, or polyploid. Here we can see if there is a single chromosome, there are 3 chromosomes, the cell is called as haploid. If each chromosome is in the form of duplicate, then the cell is called as diploid. So there is a single copy chromosome, then it is called as haploid cell, and if there are 2 chromosomes mean in the form of homologous chromosomes, they will be called as a diploid. The number of genes on chromosome is vary between species to species. Here if we will see that in case of humans there are approximately 25,000 to 27,000 genes are present on the human chromosomes, while in case of rice there are approximately 45,000 genes are present on the chromosomes. In case of Drosophila, there are approximately 13,700 genes are present on the chromosomes of the Drosophila, and on yeast there are approximately 5,800 genes are present on the chromosomes of the Drosophila. So the number of genes on the chromosomes vary between species to species. Prokaryotic have higher gene density as compared to eukaryotes. In case of prokaryotes, they have high gene density. Genes are placed side by side, while in case of eukaryotes, there is a distance between the genes and gene density is less as compared to prokaryotes. Here we can see that in case of prokaryotes, the size of the genome in case of prokaryote is less, while the size of the genome in case of eukaryotes is large. And on the other hand, if we see approximate number of genes in prokaryotes is less, while the number of genes they are maximum in eukaryotic cells. In case of vertebrates and in case of plants, they have higher number of genes as compared to prokaryotes. If we do the comparison between the prokaryotes and eukaryotes based on the gene density, so gene density is higher in prokaryotes, where we can see that there are approximately 801,000 genes per MB of DNA, while in case of vertebrates and in case of plants, we see there is a range between 20 to 100 genes per MB of DNA. If we make a comparison between the genomes of prokaryotes and eukaryotes, prokaryotes have less size of the genome as compared to eukaryotic genomes. If we do a comparison based on the number of genes, prokaryotic genome has less number of genes, while eukaryotic genomes they have large number of genes on their genome. While if we do a comparison based on the gene density is large in case of prokaryotes, while gene density is less in case of eukaryotes. In case of prokaryotes, approximately 800 to 1000 genes are present within 1 MB of DNA, while in case of plants and vertebrates, there are approximately 20 to 100 genes are present within 1 MB of DNA.